Hello everyone, welcome to Invice Academy. In today's session, we are going to discuss about subcellular structures of prokaryotic and eukaryotic cells. As we discussed before, there are mainly two types of cells. One is prokaryotic cells and another one is a eukaryotic cell. Prokaryotic cell, as its name indicates, it is a primitive cell, or we can say it as it is a very simple form of a cell. Eukaryotic cells, it is a complex form of a cells with lots of internal organelles. As I said before, it is prokaryotic cells. It is a simple form of a cells which have a membrane bound, which is lack membrane bound organelles. Whereas the eukaryotic cells, it is a more complex form of a cells and contain distinct organelles for a specialized functions. So here is one table. It is just we are doing a comparison between subcellular structures of prokaryotic and eukaryotic cells. Here you can see that in case of size, prokaryotic cells are more smaller, that is, it ranges from 1 to 10 micrometers. Eukaryotic cells, it ranges from 10 to 100 micrometers. If you if you are saying about the genetic material, that is a nucleus. In prokaryotic cells, there is no existence of a true nucleus. Whatever the genetic material present in a prokaryotic cells, it may be a DNA or a RNA, it is located in a nucleoid region. Nucleoid region is nothing but in prokaryotic cells where this genetic material is present, that particular region we call it as a nucleoid region. In eukaryotic cells, there is a true nucleus, it exists, which is a membrane bound organelle. Inside that, the genetic material is protected or preserved. If you are talking about membrane bound organelles in prokaryotic cells, membrane bound organelles are absent. In case of eukaryotic cells, there are lots of membrane bound organelles are present with which are doing different functions. If you are talking about DNA or a genetic material in prokaryotic cells, single circular DNA is present, and it is there is no histone proteins are present. What are these histone proteins? As we will discuss it later. And in eukaryotic cells, it is a linear DNA. Remember it. If you are discussing about a genetic material, prokaryotic cell, it is having a single circular form of a DNA. It is a circular form. In case of eukaryotic cells, it is a linear form of a DNA. It is not a circular. Okay, and it is linked with the histone proteins, remember this name histone, I will explain it. And next comes to the ribosomes are the organelles or protein organelles which is present in every cells, like in both prokaryotic cells and eukaryotic cells. It is very much required for the genetic material to express in the form of a protein. That is what the work of this ribosomes. If you are checking, or if you are discussing the ribosomes in a prokaryotic cell, then whatever the ribosomes present in a prokaryotic cells are the 70s type of ribosomes. In eukaryotic cells, you can observe 80s type of ribosomes along with the 70s type of ribosomes. Why? Because usually there are two types of ribosomes are present. One is 70s type of ribosomes and another one is 80s type of ribosomes. Usually eukaryotic cells, that is advanced cells or developed cells, they are having 80s type of ribosomes, and primitive cells, they will have, as we discussed in origin and evolution chapter, this prokaryotic cells, they will have a 70s type of ribosomes. So remember this. But in eukaryotic cells, it have both 80s and 70s. Why? Because according to the endosymbiosis theory, as we discussed in our evolution chapter, mitochondria and chloroplast cells, they are formed as an organelle because of the engulfment of the aerobic and photosynthetic bacteria by the larger prokaryotic cells. And as the time passes, as the time passes, this these two bacteria, which is engulfed by that big prokaryotic cells, they converted as organelles, and it is then continued as an organelle for a respiration purpose, that is an energy generation purpose, and for the photosynthetic purpose. If you are talking about the cytoskeleton in a prokaryotic cell, it may be limited, it may be present. If it is present, then it is very much limited, or it is completely absent in a prokaryotic cells. And if you are talking about the eukaryotic cells, well developed cytoskeletons are present, which will give us structural support and involved in a intracellular transportation, which will give a structural support and intracellular transportation. If you are talking about the cell division, how this will multiply, as I said, the example of the prokaryotic cells or a prokaryotic organisms are bacteria. So, individual cell is an organism. It is nothing but organisms. Bacteria, it is having only one cell, that is prokaryotic kind of cells, and we can call that organisms as a prokaryotic organisms. So, here cell division is nothing but in case of prokaryotic organism, cell division, it is nothing but cell multiplication. But in case of eukaryotic organism, cell division, it will happen to increase the mass of the tissue, or to or for the growth purpose, or for some other metabolic requirement. That's why because of for the different purpose, this cell division will happen, and it is not a multiplication process, or it is not a reproduction process. Binary fission and mitosis and meiosis, I'll explain this also. Example for this binary fission or prokaryotic organisms are bacteria and archaea, what we are discussed before. Bacteria are a prokaryotic organisms, you know that, and archaea, these are the kind of bacteria which is very much related to, very much similar to, or very much almost, it is very nearer to the eukaryotic organisms, but we can't consider it under eukaryotes because it is not having complex cells, but it can't be included under a prokaryotic cells also, because it is having advanced organelles compared to the, or advanced metabolic uh, pathways compared to the normal bacterial group, because of that only it is able to survive under extreme conditions. Eukaryotic cells examples, the organisms which are having eukaryotic cells are plants, animals, fungi, and protists. All these four groups are comes under eukaryotic organisms, and it have, and all these four group organisms, it have a eukaryotic cell. Okay. Okay, this is how our prokaryotic cells will look like. It is having a simple one outer covering that is called that is made up that is either a plasma membrane. There are two types of cover or covering it is having. One is a plasma membrane, that is the innermost covering, and the cell wall, that is the outermost covering. And inside material, this green color liquid, it's a liquid or a gel kind of thing, it is called as a cytoplasm. And there is a one more in between these, as a capsule is present, and this is a genetic material which is not have any nucleus, it is not having any nucleus, and it is not covered or a protected organelle. It's simply a colloid, it is, and it is also have some foldings in the plasma membrane region. That foldings we will call it as a mesosomes. The foldings in a prokaryotic cells we call it as a mesosomes, and it have a ribosomes. We know this it is a 70S type of ribosomes are present in prokaryotic cells, and it is having a flagellum. Flagellum are the flagella. It helps in a movement. And one more important thing is, in a prokaryotic cell, in a bacterial cell, along with the genetic material, this is a main circular DNA. There is a main genetic material. Along with a main genetic material, there are small circular form of a DNA is also present. This is called as a plasmids. It is a small circular form of a DNA. It involved in a many metabolic pathways, and it will give a resistant power, or it will give a separate properties to that particular bacteria. That's why these plasmids are present inside the prokaryotic cells. And one more thing what we need to remember is, these plasmids, it will replicate independent of this. That means in a single prokaryotic cell we'll have only one genetic material, that is only one circular DNA we are having, which is super coiled, and it will look like this. And this region, this dark region, we will call it as a nucleoid. And in a single cell there are lots of plasmids were present depending upon the requirement. It may ranges from 20 to 100. And this is what I'm telling last time about the histones. As I said, in prokaryotic cells there's a single genetic material is there, but it is not bounded with a histone proteins. But in case of a eukaryotes, it is along the genetic material is bounded with a histone proteins. This is a one point what I mentioned when we are discussing about the difference between these two cells. And here you can see that in the right side, this is how our genetic material or eukaryotic genetic material will look like in a condensed form. We'll call it as a chromosome. See, this is a simple form of a DNA. It's an open structure. That's a double helix model, what we'll call. This is how our DNA will look like if it is opened and zoomed properly. So this DNA is negatively charged molecule. DNA is always a negatively charged one. So these histone proteins, these yellow color round shaped structures, this is a histone proteins. There are mainly, here you can see that there are mainly 5 types of histone proteins are present. There are mainly 5 types of histone proteins are present. That is, you can see it here: H2A, H2B, H3, H4. H2A, H2B, H3, H4, and one more histone proteins it is present here, H1. So there are mainly 5 types of histone proteins: H1, H2A, H2B, H3, H4. These 5 proteins, we'll call it as a histone proteins. What happens in a eukaryotic genetic material is, this negatively charged DNA, it rotates, it circulates around this histone proteins. It is in octamer form. That means these 4 proteins, that H2A, H2B, H3, H4, it will be in a duplicate form. That means in one this kind of structure there are 2 H2A are present, and 2 H2B are present, 2 H3 are present, and 2 H4s are present. But this H1 we will call it as a linker histone. Okay, this one we call it as a linker histone. It will be present in a single form. It is not in a duplicate form. It will present in a single form. It will connect this, it will hold this octamer together. This is what the function of this H1. This is the 5 types of histones. This is, so see, this is one octamer, this is one octamer, this is one octamer. So around this octamer our DNA will coil. It will coil around that octamer form, and this octamer, so this is how it will be. Then it will further coil together. Then it will form a chromatin fiber. These chromatin fibers further it will coil around it, and it will form a condensed form of a DNA, that is a chromosome. This is how it present in eukaryotes. In case of a prokaryotes, there is no presence of, there is no histone proteins are present. There is no histone proteins are present, remember, but non-histone proteins are present in a prokaryotic genetic material, which will hold this genetic material together in a nucleoid region. So what are the examples? It is not important, but you can just try to remember. It is a HSF, FIS, HU, HNS, SMS. These are some kind of histone proteins, non-histone proteins, which are present in a eukaryote. The important point to remember is, in case of a eukaryotes, there are histone proteins which will hold genetic material together and a condensed form, and it will form a chromosome. In case of a prokaryotes, there is no histone proteins are present to form a coiled structure. And one more thing to remember is, in case of prokaryotes, we have a circular form of a DNA. In case of eukaryotes, we will have a linear form of a DNA. And this is what I am saying about. There are mainly two types of ribosomes. Like one is 70S type of ribosomes, and one more is 80S type of ribosomes. Usually this one ribosome, that if you're taking 70S ribosome, if you're taking, this is made up of two subcomponents, that is 50S subunit and 30S subunit. If it is comes together, 50S subunit, there is this one bigger one, and this small 30S subunit, if it is comes together, then it will form 70S type of ribosome. This is what present in a prokaryotic cells or a primitive cells. In advanced or complex eukaryotic cells, 80S type of ribosomes are present. That means in 80S type of ribosomes, the bigger part we will call it as a 60S subunit, and the smaller part we will call it as a 40S subunit. This is the difference. So, if it is a complex or a eukaryotic cell, 80S type of ribosomes are present in a cytoplasm, but in mitochondria and in chloroplast, because it is these two organelles are from prokaryotic cells. So here in these two cells we can observe 70S type of ribosomes. Otherwise, only in prokaryotic cells we can observe this 70S type of ribosome. And this is the one more thing what I am discussing. In case of prokaryotes, what I said, the mode of cell division, that is a binary fission. This is what I said. In case of in case of eukaryotes, that is either mitosis or meiosis, depending upon the requirement. That is what will happen there. And one more thing I said, in case of prokaryotes, if cell division is happening, means that is the process of reproduction or a cell multiplication. So this is how this binary fission will happen. It is a very simple procedure. This is the parental cell, and suddenly this, if it is a time of binary fission, means suddenly elongation of this nucleus will happen, and at the same time, after elongation, these two nucleus will be separated. See, and either side it got separated, then division of the nucleus in the cytoplasm happen, then division of the cytoplasm will happen. So hence it forms a two daughter cells. What is the result of these two division? This division, it will form a two daughter cells. This is how binary fission will happen. This is the in eukaryotic cells, the mode of cell division is either mitosis or meiosis. That is what I said that time. So mitosis, usually if a plant, if an organism wants to grow, or some repair things is happening on its body, that time mitosis process will happen. Meiosis process will happen only during the reproduction time, and we will discuss this whole process, how it will happen, everything in our coming classes. This is a basic difference. Just try to remember, in your prokaryotic cells, binary division is the mode of cell division, and in case of eukaryotes, either mitosis or not either, mitosis and meiosis, these two are the okay. Now you know the basic difference between prokaryotic cells and eukaryotic cells. Now we will we will see in brief about the prokaryotic or a subcellular structure of a prokaryotic cells, how it will look, what it is made up of, and what is the function of that particular organelle or a particular content in the cell and what it will do. So, if you are saying about the prokaryotic cell, the outermost cover of the cell, that is a cell wall. Then, how it is made up of? The composition of this cell wall is peptidoglycan in bacteria. Remember it, in bacteria, the cell wall is made up of peptidoglycan layer. Peptidoglycan layer. In case of archaea, it is made up of pseudopeptidoglycan layer. Composition of a cell wall, it is made up of peptidoglycan layer, and in case of archaea it is made up of pseudopeptidoglycan layer. Then what is the function of this cell wall? This cell wall, it provides a shape, particular shape to the bacteria, or it will protect it from the extreme conditions or outer surroundings, and structural integrity. It will provide shape, protection, and structural integrity to that particular organisms. And inside this cell wall, this is the outermost cell. Okay, cell wall is the outermost cell, and inside this cell wall there is one more membrane is present, one more covering is present, that is plasma membrane. Okay, the second cover, that's a outermost cover is cell wall, next to that plasma membrane it will be present. So what it is made up of? It is made up of phospholipid layer. Okay, it is made up of phospholipid layer embedded with proteins. This phospholipid layer, that layer, it is embedded with proteins. Okay, what is the function of this particular layer? This regulates the transport of the substances in and out of the cells. So, if some particular substance need to be transported out of the cell or it need to be come into the cell, then this is the substance, this is the membrane, which is we'll say it as it's a live membrane. Okay, because it can differentiate between the things. Okay, whether it can differentiate and it can judge the things, like whether that particular thing should come inside or it should go outside. It will decide based on the transporters present on this. But cell wall, there is no transporters are present, nothing is there. Cell wall, just it will give a protection. This is how bacterial cell wall will look like, this above portion. And based on a cell wall structure, basically there are two types of bacteria. We can differentiate whole group of bacteria into two types, that is gram positive bacteria and a gram negative bacteria. You may heard about this. Like in gram positive bacteria, here you can see the outer membrane. This is the inside part of the bacteria. This is the membrane. So that is a cell wall, and this is a plasma membrane, that what I said, like this is the second layer, and this is the outermost cell wall. This is from here to here, it is a cell wall. See here, outer membrane, it is made up of a peptidoglycan. That is what we discussed. This is a the purple color one is a peptidoglycan layer, and inside this, for between this peptidoglycan and plasma membrane layer, there is a space, there is a small space it is present, that is called as a periplasmic space. It's not important, like just remember, in a gram positive bacteria, the cell wall is made up of only peptidoglycan. This one. In case of a gram negative bacteria, you can see here we have only one layer of this peptidoglycan, and above that there is some lipoproteins are present, and there is lipopolysaccharides are present here, and there is the outermost membrane. There is one more membrane is present. One more cytoplasmic membrane was present on the surface. Above this, above the cell wall layer, there's a peptidoglycan layer, there is one more membrane was present, and in between some porins are present, that is a lipoproteins. Those are transporters are present. There is an extra one layer. Because of this, it is called as a gram negative bacteria. Because of this, it is called as a gram negative bacteria, because Because of this, it will give a negative result in a gram staining. Why this name comes like gram positive and gram negative means, during a gram staining it will not a stain by itself, because of this layer, whatever the stain we are adding to that, it will be washed out while during washing. But in case of a gram positive bacteria, this peptidoglycan, it will absorb the stain and it will give a, it will be observed as a blue colored cells. Because of that it is named as a gram positive and gram negative bacteria. The simple thing, there are two types of prokaryotic bacterial cells. One is a gram positive and gram negative. In a gram positive bacteria, it is only the outermost layer, it is only made up of peptidoglycan. But in case of a gram negative bacteria, along with the peptidoglycan layer or over that peptidoglycan layer, there is one more outer membrane will be present. And in case of archaea, this is archaeal cell wall. This is archaeal cell wall. Usually in see, this is a archaeal cell wall. This is how it will look. Usually in a membrane layer, usually there will be a ether linkage. But in case of a archaea, there will be a ether linkage. But in case of a normal cell wall or a bacterial cell wall, you will observe ester linkage. This is a small difference. There's only one difference between archaeal cell wall and a bacterial cell wall. That is, in case of archaeal cell wall, ether linkage will be present between the things, and in case of bacterial cell, ester linkage will be present. And this is the second layer, that is a phospholipid layer, or we can also call it as a plasma membrane, and we'll also call it as a semi-permeable membrane. We'll also call it as a semi-permeable membrane. Why it is called as a semi-permeable membrane means, it will not allow everything. If one particular cell is allowing everything to come in, then it is a permeable. Okay, if it is not allowing nothing, like a cell wall is not allowing anything, so that is a, it is not permeable membrane. If it is allowing the selected things or particular things, then we can call it as a semi-permeable membrane. So phospholipid layer or a plasma membrane, it is called as a semi-permeable membrane. And I said before, it is made up of a phospholipid bilayer, phospholipid and proteins, embedded proteins. That is what we discussed. And here you can see that these are the these two, like this is a phosphate. Why phospholipid bilayer means, this is the one layer up to here. See, this is one layer. This is phosphate group, and this is the fat group, lipid layer. This is phospholipid. So one layer. This is a phospholipid. That's why two are there. That's why it is a bilayer, phospholipid bilayer. And these are the proteins. Embedded proteins are present. This is what we discussed. These are the embedded proteins which are present on the plasma membrane, and these are the proteins which allow, see, it will look like this, there will be a space inside, and these are the proteins which allow the some particular compounds to come inside or go out. It will select the things. See, maybe it may be selectable to the Na plus, or this one it will allow only K plus, and so individual protein, there is a in different functions are present for individual protein, different functions are present. So individually it will allow the different compounds to, it will select that compounds, and it will then it will allow those particular compounds to come in or go out. And one more thing is like some carbohydrate chains are also present on the phospholipid layer, but main component of the phospholipid bilayer is phospholipid layer along with the embedded proteins. Okay, up to this point we studied about the outermost structures in bacteria or in a prokaryotic cell. The outermost layer is a cell wall, and the second layer is a plasma membrane. Cell wall, it will just give a protection, and plasma membrane, it will help in a transportation of a substances. Then comes to then we'll go to the internal part. What is there inside those two membranes? There is a cytoplasm. Cytoplasm, we can say that it is a gel-like substance where cellular process can occur. It is a gel or a gel kind of substance. It is present throughout that cell. See, in this image, this light blue color, whatever you are seeing, this is, we are calling it as a cytoplasm. Cytoplasm is a gel-like substance which is present inside that particular membrane. So inside this, okay, in that particular cytoplasm, many organelles or a nucleus or a genetic material, it is embedded. So inside those two membranes, there is a cytoplasm. Okay, gel-like substances where cellular process can occur. That is what we saw now. And what are the components in case of prokaryotes? What are the components may be present inside that or embedded in that cytoplasms are like it, as we said before, ribosomes are present. Different enzymes will be present and nutrients. Whatever the nutrients it taken in, that will be present in that ribosomes. Next part of the prokaryotic or next organelle or a next a component of the prokaryotic cell, that is a nucleoid. This is a region containing a single circular form of a DNA. The main thing, nucleoid is a region and prokaryotes circular form of a DNA is present. There's the two important points to remember. And function of this particular nucleoid, it stores the genetic material and controls the cellular functions. We know genetic material is present in every cell to control the metabolic process and different functions. That is what it is doing. And the fourth component of the prokaryotic cell is a ribosomes. As we discussed before, in case of prokaryotes, 70S type of ribosomes are present, and this 70S type of ribosomes, it is made up of RNA and proteins. Whatever the ribosome it is, ribosomes are made up of RNA and proteins. Remember, ribosomes are made up of RNA and proteins. Okay, but in case of prokaryotes, 70S type of ribosomes are there. Function, synthesizes a protein. And next organelle is a flagella. As we saw in that figure, flagella, it is a long thread-like structure or a whip-like structure that is made up of flagellin. Flagella, flagellin. Flagella is made up of flagellin. Flagella is made up of flagellin. Okay. The function of this particular flagella is it provides motility or it helps in a movement. And next part of the prokaryotic cells, that is pili or a fimbriae. Pili or a fimbriae. What is the function of this pili? It involved in a DNA transfer during a conjugation. Okay. In case of prokaryotes or in case of a bacteria, there are three mode of DNA transfer. Okay. In case of prokaryotes or not in prokaryotes, in case of a bacteria, there are three types of DNA transport. DNA transfer. Okay. One is transformation. One is transformation. Second one is conjugation. And third one is a transduction. Okay, in bacteria there are three types of DNA transfer: transformation, conjugation and transduction. And this pili, if that pili is present in that particular bacteria, then the mode of transportation, then mode of DNA transfer in that particular bacteria are conjugation. So how this pili is involved means, see, if these pili are present, it's a small, okay, it looks like little bigger compared to the fimbriae. These small hair-like structures are a fimbriae, and a little bigger structure it is a pili, and far bigger structure it is flagella. This is how it will look like. So this is a bacterial cell. As I said, the longer thread-like structure, it is a flagella. It helps in a movement, and the small hair-like structures, it is a fimbriae, and the little longer structure, it is pili. Usually, another bacteria will come, which is also have this pili. It will come and it will attach to this particular bacteria through this pili, and it is having genetic material inside, and it is having its genetic material inside. Okay, and through this pili, this particular genetic material, it will pass to this. So this is how it will happen. And this fimbriae, that small hair-like structures, it involved in a it helps in adhesion to one particular surface. This what's the function of this fimbriae. And inclusion bodies, the next component of the prokaryotic cell, that is the inclusion bodies. It is a storage granules for the nutrients like glycogen, for polyphosphate, or a sulfur, whatever the nutrient. As I said, for because as I said, some prokaryotic cells still it will have a chemolithotrophic kind of food habit. Because of that, the mode of nutrition is different. Some bacteria, they still today they will depend upon H2S and iron for its for a source of a nutrition. So depends upon the nutrient requirement, it will intake its nutrient, and it will be stored in the form of a storage granules, and small granule kind of granule structures are we can observe inside this bacterial bodies or prokaryotic cells. That will call it as a inclusion bodies. And function of this inclusion bodies is to store the, athwa it will act as a energy reserve, and it will energy reserve material, that like that we can see. And one more is a plasmid. It is a small circular extra chromosomal DNA molecules. As I said, in one particular prokaryotic cell, there is a genetic material. There's only one genetic material will be there. It is only one, and there are, depending upon the type of the prokaryotic cell and depending upon which bacteria it is, there is a presence of a plasmids. In some prokaryotic cells, there is no presence of plasmids. There will be no plasmids are present in that particular cells, but in some particular bacteria you can see up to 100 numbers of plasmids, and in particular cells you will observe only 10 it observe it in a 10 numbers. So depends upon the type of the bacteria and its requirement, number of plasmids will be present. Plasmids are the, remember the definition of this, like it is a small circular extra chromosomal DNA. If in some questions, if they ask what are the extra chromosomal DNA present in the prokaryotic cells, that is a plasmids. It carries a genes for antibiotic resistant and survival advantages it is having. So as I said, it will these plasmids, it will replicate or multiply independently without the guidance of this genetic material, independent to this genetic material. So it will not depend upon the genetic material to replicate, so it will replicate individually. That's why many copies of plasmids are present in a particular cells. And it will give a antibiotic resistance. So if some bacteria are resistant to some particular antibiotics, or it is surviving in an extreme condition, that particular gene may be present in this particular bacteria, or sorry, not in a bacteria, in a plasmids. So this is how a bacterial plasmid or a plasmid will look like. As I told before, it will replicate independent of this genetic material. Because of that, this is how it will look. If you do the mapping of the plasmid, it will look like this. It is having its own origin of replication. That means it helps in, it starts the replication, and it is having the antibiotic gene somewhere, and if it is there, then it will give the antibiotic resistance, and it is having the different parts, it is having different parts independent of the genetic material. So These are all the components which are present in the prokaryotic cells. This is the overall structure of the prokaryotic cells. As we discussed, the outermost layer is a cell wall. Then comes a plasma membrane or a phospholipid bilayer or a semi-permeable membrane. That one also we can call it as. And then inside this green, that is a gel-like substance, that is a cytoplasm. In that, many different components are embedded. That includes a genetic material, and some inclusion bodies also present. That is what we studied, that is a storage granules. Some storage granules also will present inside this, and ribosomes are present, which will help in a ribosomes. There is a formation of a protein, it involved in a protein formation, and it is made up of RNA and cytoplasm, and some foldings of the plasma membrane. Remember, foldings of a plasma membrane, it is a plasma membrane. Foldings of a plasma membrane, it is called as a mesosomes. Mesosomes are a foldings of plasma membrane. And long thread-like structure, if it is present, that is a flagellum, it will helps in a locomotion. And a small hair-like structure, if it is present, then it is a fimbriae helps in a attachment. And a little long structure, if it is present, then it is a pili, it involved in a conjugation process. That is a DNA transfer, method of DNA transfer in a bacteria. Okay. Then after knowing about the prokaryotic cells, now we'll discuss about the subcellular structures which are present in a eukaryotic cells. So, uh, in case of eukaryotic cells, there is no cell wall. Eukaryotic cells, it will not have any cell wall structure. The first or outermost layer of a eukaryotic cell, the outermost layer of a eukaryotic cell is a plasma membrane. There is no presence of cell wall. Cell wall is absent in eukaryotic cell. Then what is the function of the plasma membrane? As we discussed in the while explaining this prokaryotic cell wall structures or structure, this plasma membrane, it is made up of phospholipid layer with the embedded proteins, sterols like cholesterol, and sterols like cholesterol, that is a carbohydrates which is present on that. So plasma membrane, remember, it is made up of phospholipid bilayer which are embedded with proteins and sterols like cholesterol, that is the starch. So carbohydrate which is present there. What is the function of this plasma membrane in eukaryotic cell? Same thing, it regulates the material exchange and protects cellular integrity. In case of prokaryotes, cell wall, it maintains the structure and it is maintaining the cellular integrity. But in case of a plasma membrane, in case of eukaryotes, plasma membrane, this is the outermost layer and it is involved in material exchange as well as protection. And then comes the important organ, that is a nucleus. So nucleus, usually it is enclosed by a double membrane layer that is called as a nuclear envelope, that is called, we'll call it as a nuclear membrane. Okay, nucleus, it is having a double membrane layer. This is also phospholipid layer itself. See, this is the cell. Inside that cell, nucleus is present. It is a one organelle. This is covered by a membrane, phospholipid layer, and inside this one organ is present, nucleus. It is this individual organ. This organelle is also covered by a phospholipid membrane, and this membrane, it will have a pores in between. So this is how it will look like. There will be a pores. That pores are, we'll call it as a nuclear pores, and inside this nucleus we will see a genetic material, that is a DNA plus proteins, and inside this one more organelle is present, that will call it as a nucleolus. Okay, and see, what is the function of this nucleus? It stores the genetic material and regulates the gene expression. And nucleolus, it is main function of this nucleolus is to produce and assembles the cell ribosomes. So it controls the ribosome assembly. Okay, this is how nucleus will look like, and this is the nucleus. And this is here black pore kind of structure. What you are seeing, these black spots are pores which are present in this nuclear membrane. This purple color is a nuclear membrane. Inside this some liquid kind of structure is there inside this nucleus, that we call it as a nucleoplasm. And all our genetic material, it is present in this nucleoplasm. In this nucleoplasm our genetic material is present, that is a DNA. And inside this one more round shaped structure is present, that is nucleolus. This is where this nucleolus, it controls the ribosome production and assembly. And this exactly immediately next to this nucleus there is endoplasmic reticulum is present, that is a one more organelle. So it will look like this. See, if you are seeing this whole eukaryotic cell, this is how eukaryotic cell will look like. It consists of lots of cellular organelles, like which will do different functions. And this is how it is organized. The outermost cell, that is a plasma membrane, is present, and inside this then some round structure you are observing. This is nucleus and this is nucleolus, and immediately next to that endoplasmic reticulum is. Okay, coming to the next part of the prokaryotic cell, that is a cytoplasm. As we discussed in a as we discussed while we are discussing about the prokaryotic cell, here also cytoplasm, it is a gel-like matrix containing all the organelles and a cytoskeleton. Here remember, prokaryotic cell, eukaryotic cells, it have a different organelles and a cytoskeleton. I'll explain what is a cytoskeleton in the next part. And so here this cytoplasm, it is embedded with different organelles and cytoskeleton. And what's the function of this cytoplasm here? It is a site of metabolic activities and organelle functioning. So it will hold the organelles in a proper place, and it will involve in a metabolic activities, as we discussed in a prokaryotic cell structure, as we discussed while explaining or discussing about the prokaryotic organelle structures. The next organelle present here, it is a ribosome. It's a clear difference what we are discussed. Like here, ATS type of ribosomes are present in the cytoplasm, and 70S type of ribosomes are also present in the eukaryotic cells, but inside the mitochondria and chloroplast. So usually if someone asks, or if some question arises, like they asked in an exam, like what is the type of ribosome present in a eukaryotic cell, then we should choose ATS type of ribosome. And if they are extending the answer, then we can say 70 ATS type of ribosome in a cytoplasm and 70S type of ribosomes in a mitochondria. But if it is a simple question, what is the type of ribosome present in a eukaryotic cell, then it is ATS type of ribosomes. And these ribosomes, they are involved in a protein synthesis. Then comes to the next organelle, that is mitochondria. As I said, these mitochondria are present inside that eukaryotic cells due to the engulfment, because it is engulfed by the ancestral bacteria to the present bacteria or a present eukaryotic cell. Uh, so mitochondria, it is also a double membrane structure. It is also a double membrane structure like nucleus, and inner membrane forms a cristae. That means there is some foldings. I'll show the image. And it usually involved, what is the function of this mitochondria? It generates ATP via oxidative phosphorylation. Remember this point. In a mitochondria, energy generated, ATP will be produced, or energy will be produced. Usually ATP is the source of energy. ATP, we'll call it as a energy currency of the cells. ATP is a energy currency of the cell. So in cells, ATP is the form of the energy which will be exchanged. So if energy is produced in the body, if someone says energy is produced in the cell, then ATPs are produced. It means ATPs are produced in that particular cell. So energies are produced in a ATP. ATPs are produced in a mitochondria via oxidative phosphorylation. Remember this, it is a oxidative. Remember this word, oxidative phosphorylation. This is how mitochondria will look like. It is a double, as I explained before, it is a double membrane containing organelle. That means there is a two membranes. One is outer membrane, it is called as outer membrane of mitochondria, and this is a inner membrane of a, this is the inside. One more membrane is present, that is the inner membrane of a mitochondria. In between these two membranes there is a space. In between these two membranes there is a space that is called as an intermembrane space, and these foldings of the intermembranes we will call it as a cristae. Foldings of intermembrane, it is called as a cristae, and whatever the gel like structure present inside this intermembrane, that is called as a matrix. And as we said before, or as we discussed before, inside this intermembrane, inner membrane, there are the next part. There is a one more is as we discussed before, that's a DNA. Each and every in one particular eukaryotic cell there is only one mitochondria will be present. Not only one mitochondria, there are more mitochondria will be present, and individual mitochondria, it is having its own DNA, that is own DNA, and ribosomes, that is 70s type of ribosomes which are present inside this mitochondria. And but in a cytoplasm, as we discussed, cytoplasm, there is a 80s type of ribosomes are present. And DNA, it is having a self-replicating DNA. It will replicate independent of that of the genetic material which is present in that particular cell. Coming to the next organelle in a eukaryotic cell, that is an endoplasmic reticulum. As we discussed, or as I explained before, it is very adjacent to that of the nucleus. And there are two types of endoplasmic reticulum which is present in a cell, that is a rough endoplasmic reticulum and a smooth endoplasmic reticulum. Rough endoplasmic reticulum, why this name? Because above this endoplasmic reticulum, so many ribosomes they are sitting on that. So because of this, it will look like a rough structure. That's why it is called as a rough endoplasmic reticulum. Mainly, this rough endoplasmic reticulum, it involved in a protein synthesis and its modification. And coming to the smooth endoplasmic reticulum, where ribosomes are not present over that particular structure, then these are called as a smooth endoplasmic reticulum. And these endoplasmic reticulum, they are involved in synthesis of lipid and detoxification of a chemicals. It is involved in synthesis of a lipids and detoxification of a chemicals. The main two things remember in this organelle, that is there are two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum, it contains ribosomes on that, so it is involved in a protein synthesis. And smooth endoplasmic reticulum, it lacks ribosomes, so because of that it involved in a lipid synthesis and detoxification of a chemicals. The next, this is how endoplasmic reticulum looks like, and this small dots present on a endoplasmic reticulum structure, this is a ribosome. These are ribosomes. Because of the presence of these ribosomes on the surface of this endoplasmic reticulum, this portion of the endoplasmic reticulum, we can call it as a rough endoplasmic reticulum. And here this structure, this surface, it looks smooth because it lacks in ribosomes. So this is called as a smooth endoplasmic reticulum, and it is involved in lipid synthesis. And these portion or this part of the endoplasmic reticulum, it involved in protein synthesis. And here also foldings are present inside this, and these foldings are called as a cisternae. This foldings of a endoplasmic reticulum, it is called as a cisternae, and this space is called as a cisternal space. The next organelle of the eukaryotic cell, that is Golgi apparatus. The structure, how it looks like, it is a stacked structure, and it is also a membrane bound organelle, and it is also have a membrane bound cisternae is present. That means there is a foldings inside that. So because that portion here also we'll call it as a cisternae. What is the function of this Golgi apparatus? Mainly it involves in modification, sorting and packaging of proteins which are produced by these endoplasmic reticulum. Usually it will present adjacent to the endoplasmic reticulum. So it involved whatever the proteins which are produced by the endoplasmic reticulum, it will be transported to the Golgi apparatus. In Golgi apparatus, it will modify the proteins, then it will sort those proteins, and it will pack the proteins, and it will send to the different places where those proteins are required. This is the structure of the Golgi apparatus. So as I said before, it is also membrane bound structure, and here also you can see these foldings here. What we are seeing, this is called as a cisternae, and the space inside this is called as a lumen of Golgi apparatus. And this is the part here endoplasmic reticulum will be there. So it will produce proteins. These proteins will enter into this Golgi apparatus, and this Golgi apparatus, inside this it will do the sorting and modification and packaging. How it will do packaging? See here, you can see that formation of a new vesicles. That means whatever the vesicle which are present in a cells, or how it will transport the particular proteins, that is through a vesicle. Whatever the vesicles which are present in the cells, it is just a part, it is a part of this Golgi apparatus, or we can say that it is a sprout of that Golgi apparatus. Okay, then these particular sprout or vesicle, it will get separated from this Golgi apparatus, and it will form as a secretory vesicle, and this secretory vesicle, it will transport the protein through the particular place where it is required. The next part or a next organelle of the eukaryotic cell, that is lysosome. Lysosomes, it is also called as a suicidal bags of the cells because it contains hydrolytic enzymes. If it is leaked or if it is burst open, then whole cell will die. Because of that, lysosomes are also called as a suicidal bags of the cell. It contains hydrolytic enzymes. Remember, lysosomes are the organelles which contains hydrolytic enzymes, and it is also a membrane bound organelle, but it is a single membrane bound organelle, and it contains a hydrolytic enzymes, and if it is burst open, it will destroy whole cell. That's why it is called as a suicidal bag. And main function of this particular organelle, it degrades the cellular waste and a foreign material. If any foreign material it will enter into the cell, it will be digested by the hydrolytic enzymes which are present inside the lysosomes, and it will also degrades the cellular waste. This is the structure of the simple structure of the lysosomes. It is covered by a single membrane covered structure it is, and inside this hydrolytic enzymes are present, and to transport these hydrolytic enzymes from one place to the other, or to transport the foreign substances from outside, this is a cell, from outside the cell to the inside, there will be a transporters here. Okay, there will be a transporters will present here, so it will transport foreign material or any substance which need to be degraded it will be sent inside to this cell, and it will be degraded inside this cell, inside this organelle. The next organelle, that is a peroxisomes. Peroxisomes, it present in both plant and animal cells, it will present, but remember lysosomes, it present only in animal cell, not in a plant cell. And peroxisomes, the structure, it is also a membrane bound organelle, but it contains oxidative enzymes. Remember lysosomes, it contains hydrolytic enzymes, but peroxisomes, it contains oxidative enzymes. The function of this peroxisome is very simple, that it breaks down a fatty acid and detoxifies the harmful substances. Okay, the main function of the peroxisomes are, it breaks down the fatty acid degradation, and it also involved in detoxification of a harmful substances. In case of plants, it do one more thing, or it involved in one more metabolic pathway, that is photorespiration. Okay, so this is peroxisomes. It is also membrane bound organelle, but it contains oxidative enzymes, and it involved in fatty acid breakdown and detoxification of a harmful substances. In case of a plants, it involved in a photorespiration process also. This is how peroxisomes will look like. It contains it is also a single membrane containing organelle, and inside this you will see the oxidative enzyme core. The center part, whether it's a crystalline core, it is nothing but a oxidative enzymes. It will present inside this, so it is a place where oxidative enzymes are present. That is why usually in an electron microscope, if you are observing this particular organelle under electron microscope, you will see this crystalline core. This is nothing but a oxidative enzymes, and it is surrounded by a single membrane. The next organelle of a eukaryotic cell, that is a cytoskeleton. Cytoskeleton, there are mainly three types of cytoskeleton. There are three components mainly. One is microfilaments, the second one is microtubules, and the third one is intermediate filaments. Cytoskeleton, mainly it is made up of three particular components. One is microfilaments, microtubules, and intermediate filaments. These microfilaments, usually it is made up of actin filaments. Remember it, microfilaments, it is made up of actin filaments. It is mainly involved in shape and motility. Shape of the cell, okay, shape and motility. So it is controlled by microfilaments. It is made up of actin. So remember these 2 points. Microfilament, that's the first component of the cytoskeleton. It is made up of actin, and it mainly controls the shape and motility of the cell. And second one is microtubule. It is made up of tubulin. See, tubes, microtubules, tubulin. Remember like that. Microtubules, it made up of it is made up of tubulins. Okay, so tube, like that you can remember. Microfilaments, actin, like that you can remember, but you can't remember this. So if you are remembering microtubules are made up of tubulin, then automatically you can remember microfilaments are made up of actin. It's a tubulin based structure for a intercellular transport and spindle formation. So this spindle formation, I'll explain it during a cell division process, that is during mitosis and meiosis. Usually spindle fibers are formed to separate the genetic material. We know it is in a chromatin form. To separate this one, usually this spindle fibers are formed. through this these chromosomes are get separated like this. This is what it is. And it usually help microtubules mainly involved in intracellular transportation and spindle formation. And third important component of this cytoskeleton, that is intermediate filaments. It provides a mechanical support. Intermediate filaments, it provides a mechanical support. The main function of this is shape, aids the movement and transport, and supports the organelle arrangement. It involves in many functions. Usually these intermediate filaments, it is made up of many things, like for example, depending upon the cell type where it is present, depending upon the cell type, these intermediate filaments, it is made up of different components like keratin, desmin, vimentin. So there are different components. It is made up of different components depending upon the cell type. This is how cytoskeleton will look like inside the cell. It is not easily visible like other organelles. It spread, these cytoskeleton, it spreaded throughout the cell. Throughout the cell, inside this cytoplasm, these cytoskeleton are spreaded, and it is having its own function. Like for example, microfilaments, it is involved in maintaining the shape. Microtubules, it involved in a transportation. See these a purple color line, this is microtubules, it involved in a transportation. And spindle fibers, remember, my microtubules, a spindle fiber and transportation. And microfilaments, it involved in a, it maintains the shape. And intermediate filaments, as I said, there are different it is made up of different components. And depending upon the cell where it is present, these intermediate filaments will involve, it will do its own work. Coming to the next part, that is vacuole. It is a very large, a single membrane bound. It is also a single membrane bound, a compartment which is present in both animal and plant cell, and it is very large if it is present in a plant cell. In case of animal cell, it is a very small structures, but in case of a plant, it is a large single membrane bound, very large structure. If you are observing a plant cell, if you are observing a plant cell under a microscope, then the main two organelle, if you are observing, that is a nucleus and a large vacuole. But in case of a animal cell, it is a very small structure. If you are observing animal cell under microscope, you will observe nucleus as usual, but you will not able to observe these vacuoles because it is a very small structure. And what is the main function of this vacuole? It stores water, nutrients and waste product of the cell. It stores water and nutrients and waste product of the cell, and it provides the turgor pressure in a plants. What is this turgor pressure? It is a pressure which is required to maintain the cell structure. The cell is a to maintain the cell shape, this turgor pressure is required. If turgor pressure is very low, if it is becomes low, the cell will shrink. So turgor pressure is the pressure which are maintaining a shape of the cell. See, this is how vacuole will look like in a plant cell. It is a very large single structure, and in case of animal cell, it is very small structures. But there are many vacuoles may be present in case of animal cell, but in case of plant, only one vacuole will be present, and it is a very large structure, and it is made up of a single layer, and this single layer, it is called as a tonoplast. The outermost layer of the vacuole, it is called as a tonoplast. It is a single membrane bound structure, remember this, and it is very large in case of plant cell, and it is very small and many in case of animal cells. Coming to the next organelle in case of a eukaryotic cell, that is a chloroplast. Remember one thing, it present in plants only. Structure, it is a double membrane structure. It contains internal thylakoid stacks. That thylakoid stack, it is called as a grana, and a stroma. Stroma is nothing but a gel-like substance which is present inside the chloroplast, that is called as a stroma. And the main function of this chloroplast is to do a photosynthesis. Photosynthesis is nothing but converting sunlight into a chemical energy. So this is how chloroplast will look like, and it is a double, as I told before, it is a double membrane bound organelle. There is a two membranes, that is the outermost membrane and a innermost membrane, and there is a space between these two membranes. It is called as an intermembrane space, and this stack like structure, this is called as a grana. All together it is called as a grana or a granum. Individual structure of this granum, it is called as a thylakoid. Okay, individual structure is called as a thylakoid. All together, the whole stack of these thylakoids are called as a granum or a grana. So these connection between these two grana, there is a small connection between these two grana. This connection, it is called as stroma lamellae. This connection is called as a stroma lamellae, and gel-like substance where these granum or a thylakoids are present, that is called as a stroma. Gel-like substance what is present inside the chloroplast, it is called as a stroma. As we discussed before, these chloroplast also, it is having 70S type of ribosomes. And during photosynthesis, this is the organelle where photosynthesis process is happening, and during photosynthesis, here also ATP will be released. ATP is releasing, and the process of ATP releasing in case of chloroplast, it is called as a photophosphorylation. But in case of mitochondria, it is oxidative phosphorylation. In case of chloroplast, it is photophosphorylation. The next part of the eukaryotic cell, that is a centriole. These centrioles, it is present in animal cells only, and it is a cylindrical kind of structure. It is made up of microtubules. While explaining microtubule, I explained two functions of a microtubule. One is formation of a spindle fibers during cell division. This is that these centrioles, it is made up of microtubules, and these microtubules, it will form a spindle fibers. With that, these chromatids, that is genetic material, will be separated. The function of these centrioles are, it organizes microtubules during the cell division. This is how centrioles will look like. It will present in the edges of the cell. If you consider this one as a cell, then these centrioles, it will present in a extreme or end of this cell, and it will form a spindle fibers. The thread-like structure what you are observing here, it is a spindle fibers, and this is our genetic material in the form of a chromosome. And during a metaphase stage of a mitosis, that is a cell division, there's a in between step of the mitosis, that is a metaphase. During that phase, these chromatids are arranged in a middle of the cell because of this spindle, with the help of these spindle fibers. And in the next step, with the help of these spindle fibers, these genetic material, that is the chromosomes, will get separated like this. So this is our centriole, and these are the spindle fibers, and this is how genetic material or a chromosome will get separated in the next step. This is how it is involved in a mitosis, that is a cell division, type of cell division in a eukaryotes. And one more thing, these centrioles, it is made up of microtubules, and it is in a this is how overall eukaryotic cell will look like. That is the outermost, it is surrounded by a plasma membrane. The outermost layer in case of eukaryotes, it is a plasma membrane. Then in the middlemost, the round shaped organelle, that is a nucleus. Nucleus, it is a double membrane structure, and inside that nucleolus is present, and nucleus is a structure which will store the genetic material. Adjacent to that, there is one more organelle, that is a endoplasmic reticulum. In endoplasmic reticulum, if ribosomes are present on that, then it is called as a rough endoplasmic reticulum. If ribosomes are not present, then it is smooth endoplasmic reticulum. These are involved in lipid biosynthesis, and endoplasmic reticulum which are having ribosomes on that, it will involve in protein synthesis. And these are mitochondria. As we discussed before, it is a powerhouse of the cell, and here ATPs are produced, that is the energy currency of the cell, that is ATPs are produced in mitochondria. And one more thing, whatever the process with which these ATPs are produced, that is oxidative phosphorylation. And in one cell, there are more than 10 to 12 mitochondria are present, and this is Golgi apparatus. This mainly involved in protein modification and its packaging and transportation in the form of vesicles. And these are the vesicles, Golgi vesicles, which need to be transported now. And these are lysosomes. As I explained, it is a suicidal bag of the cells. It mainly made up of hydrolytic enzymes. It involved in destroying foreign materials and waste product. And these are peroxisomes. These are mainly involved in fat metabolism and detoxification also. And in case of plants, it involved in photorespiration. And these fiber kind of things which you are observing in this image, these are cytoskeleton. There are mainly three types of cytoskeleton, that is microfilament. One is microfilament, second one is microtubulin, and third one is intermediate filaments. And microfilaments, it is made up of actin, and microtubules, as I said before, tubulins. And ribosomes, instead of presenting on endoplasmic reticulum, some ribosomes, it will available freely on the cytoplasm. It also involved in protein synthesis. And cytosol, it is a gel-like substance where all the organelles are embedded, and many metabolic pathways are happening in this cytoplasm. And okay, so this is the overall structure of the eukaryotic cell, and these are the organelles which are present in a eukaryotic cell. Okay, now we will see what is the key differences between these prokaryote and eukaryotic subcellular structures. It is like a glance or a remembering. We are trying just try to remember what we discussed before. If you are talking about the genetic material in prokaryotic cells, it is a single circular DNA, but in case of eukaryotic cell, it is a linear DNA. It is not a circular DNA. And it is the region where this genetic material it is present. We call it as a nucleoli. It is not a membrane bound organelle. It is simply, it is present in that particular region. That's it. But in case of eukaryotic cell, this linear DNA, it is enclosed in a nucleus. If you are talking about the organelles present in both the cells, in prokaryotic cells, different organelles, it is not there. These organelles are absent, and functions of these organelles are performed in cytoplasm. There is no separate organelles or a compartment to do these metabolic activities and all. It will done in a cytoplasm in case of prokaryotes. In case of eukaryotes, there are different organelles are present, and it is compartmentalized functions are happening there. If you are talking about the ribosomes and size of the ribosomes, in case of prokaryotic cells, it is very smaller and 70s type of ribosomes are present. In case of eukaryotes, it is little bit larger and 80s type of ribosomes are present in the cytoplasm of the eukaryotic cell. 80s type, it is present in a cytoplasm. 70s type of ribosomes are present in mitochondria and chloroplast. If the question comes like, what is the type of the ribosome present in a eukaryotic cell? Please remember it is 80s type of ribosome. If they are asking that question in deep, then they will give along with that. If they give like 80s type in cytoplasm and 70s type of mitochondria and chloroplast, then that was the answer. That is the answer. If you are talking about the cell wall, in prokaryotic cells, yes, those cells it will made up of the outermost layer of the prokaryotic cell, that is a cell wall. It is made up of peptidoglycan, and in case of archaea, it is a pseudopeptidoglycan layer is present there. But in case of eukaryotic cells, only in plants, cell wall is present, and it is made up of cellulose, not a peptidoglycan. It is made up of cellulose only in case of plants. In case of fungi also those group of organism, they also have a eukaryotic cell, and in fungi it is made up of chitin. Remember, in plants it is cellulose, in fungi it is chitin. In case of bacteria, it is peptidoglycan. In case of archaea group of organism, it is pseudopeptidoglycan. Coming to the locomotory organ or a motility part, in case of prokaryotic cells, flagella is present. If flagella is present, that is a locomotory organ of the prokaryotic cell. This flagella, it is made up of flagellin. In case of a eukaryotic cells, flagella will be there. If it is not, sometimes cilia will be there, and it is made up of a microtubules made up containing flagella or cilia is present. That is in case of eukaryotes. 9+2 arrangement is arrangement of microtubule. You can observe what is this 9+2 arrangement. This is how this 9+2 arrangement will look like. In case of eukaryotic organelles, the locomotory organ, sometimes it may be a cilia or flagella. It is made up of microtubules, and it is those microtubules or these microtubules it arranged in 9+2 manner. That means 1 2 3 4 5 6 7 8 9 microtubules in the outer side and 2 in the inner side. Because of this model is called as a 9+2 model or a 9+2 arrangement of flagella. So this is how it will look like. In overall, in today's section we discussed about subcellular structures or organelles of prokaryotes and eukaryotic cell, and some important points also I mentioned, and try to remember those points. And if you're having any doubts reading these points, then try to understand those things with the images. Thank you.